Hello, my name is Zoe Holzer. I am in Dr. Jeet Sukumaran's evolutionary biology lab, and I'm finishing my master's degree in computational science, data science. And my project is reconstructing viral dynamics of human-associated pathogen agents surrounding the COVID-19 pandemic. So the research question behind this is, how did disruption of infectious disease transmission networks in response to the COVID-19 pandemic impact the dynamics of viral populations evolving within their hosts? So the rationale is that pandemic contact rate changes should have affected contact-transmitted viruses, but clinical responses for the diseases have been studied, but not for non-COVID genomic/evolutionary responses. We use genetic sequence data under a phylogenetic framework to infer population dynamics of contact-transmitted viruses during this time, and this lets us explore evolutionary biology predictions of complex co-evolving host pathogen systems. As well, sequence data availability has increased, so potential for data mining and computational analysis should increase, and we apply this to epidemiology, where active, consistent measures for disease control are necessary. So the first objective of this project was to identify systems we could work with. So we needed timestamp sequence data before, during, and after COVID, and we used supplementary scripts to help select data sets, since like in this plot, the red line shows that for flu strain AH3N2, there was not much data during 2020 for any countries, and we also needed to eliminate data sets with poor pre-processing. So our second objective was inferring effective population size and effective reproduction numbers. So effective population size, or NE, is the number of individuals contributing genetically to a population and is usually less than the total breeding population. We inferred this with a mathematical framework called coalescent theory, where coalescent events are events where genealogies merge, and the effective population size is inversely proportional to the rate of coalescence. And we trace the ancestry of the samples from the recent samples up to the root using the time between coalescence. And we use a biceps posterior for a Bayesian method, where effective population size is sampled from the integrated out likelihood of NE, plus coalescent events given the tree times an inverse gamma prior on each temporal grouping. We sample using MCMC and model the population size over time using a piecewise linear function. And Qj and Rj in this function are complicated expressions derived from an inverse gamma prior. So effective reproduction number is the average number of secondary cases per primary case, which is used to evaluate interventions, and the goal is to get R below 1. We infer this with a framework called Variational Bayesian Skyline, or VB-Sky, and this is a scalable alternative to MCMC using optimization techniques to sample from the posterior, and it uses a divide and conquer strategy for the phylogeny. And this is the opposite, so it's forward in time. We start from the root of the tree and connect back to the recent samples. And the posterior is given here, which is the probability of the tree, and R given the data is proportional to the likelihood of the phylogeny times the likelihood of R times a hyperprior on R. And in this case, we can infer a vector of parameters, but we're just looking at R for this case. So our last objective is to test the results. So we use change point detection to test the significance of changes in NE, and we analyze both the mean and variance of the median of our inferential results, because the initial plot showed the mean of the data, or of the inference, as responsive to seasonal fluctuations. And then we also compared sample size to population size trends with cosine similarity, because we noticed some visual resemblance between the two plots. So this is future work we'd like to look at, basically seeing if the temporal distribution of samples affects the inference, and this is only some of our results, so come see my poster if you want to see the rest. Thank you.